Hello student, good morning. Today we will start the factors controlling cleavage. Yesterday we have studied the patterns of cleavage and today factors controlling the cleavage. Number 1, yolk. Type of cleavage of the fertilized egg depends upon the amount of yolk and its distribution. Types of cleavage of the fertilized eggs depend upon the amount of yolk and its distribution. The cleavage occurs more radially in the active cytoplasm than the yolk-laden cytoplasm or deutoplasm of the egg. The yolk is non-living component of the egg. Yolk is the non-living component because yolk is the food for embryo. It participates neither in cleavage nor in formation of embryo. Because it is the food for embryo, that's why it is not participate in any activity. It simply provides nourishment to the developing embryo. The yolk is present in all animals' eggs in little. Example, microlithial eggs or larger. Microlithial means little amount of yolk present in the egg. Larger example, mesolithial and macrolithial eggs. That is the large amount of yolk material present in the egg. Amounts and remains distributed evenly. Example, isolithial egg or unevenly. Example, telolithial eggs. In the ooplasm of different kinds of eggs, it has a profound deterring influence on cleavage and mechanics of moving the germ layers into their final position. The amount of yolk influences the course of cleavage by following ways. Number 1, when the amount of stored yolk increases the amount of active cytoplasm gradually decreases and the position of nucleus is variously affected in telolithial eggs, mesolithial and polylithial eggs. Such displacement of zygotic nucleus from the geometrical center of the egg to the less yolky cytoplasm is very common. The mitotic divisions of such displaced nucleus results in unequal cytokinesis and unequal sized blastomeres. Second number, every mitosis of cleavage involves movements of the cell components of chromosomes, parts of the cytoplasm constituting the chromatic figures, the mitochondria and the surface layer of the cell, the activity of which along the equator of the maternal cell leads to the eventual separation of the daughter cells. Separation of the daughter cells. During these moments, the yolk granules or yolk platelets behave entirely passively. The yolk in the uncleaved egg is more concentrated towards the vegetal pole of the egg. It is therefore at the vegetal pole of the egg blastomeres tends to divide at a slower rate, and the resulting blastomeres remains larger than those which have less yolk. When the yolk granules or platelets become very abundant, they tend to retard and even inhibit this process of cleavage. Second number, viscosity. According to various investigators, the viscosity of cortex increases before the cleavage. According to the various investigators, the viscosity of the cortex increases before the cleavage, forming a cleavage furrow as a gate-like localized thickening of the cortical layer. The cortex appears to be essential to cytokinesis. Only egg fragments containing cortical material can divide. Furrow formation can occur independently of both spindle and ester. Third number, energy requirements for cleavage. The cleavage requires a large amount of chemical energy. In the form of ATP, adenosine triphosphate, that is called as ATP molecules for its completion. These molecules are manufactured in the mitochondrial and ooplasm. During enzymatic anaerobic, that is glycolysis, and aerobic, that is Krebs cycle. Oxidation of yolk, glycogen and other energy yielding chemical molecules of egg, catabolic activity. Fourth number, organization of eggs. The influence of organization of cytoplasm on the process of cleavage is observed in the egg of eutherian mammals. The first cleavage plane divides the egg into a larger and a smaller blastomeres. The larger blastomeres then beginning to divide at the faster rate. Beginning to divide at a faster rate than the smaller blastomeres. The accelerated divisions is maintained in the daughter cells resulting from the larger blastomeres. Here then is an egg whose yolk material is at a minimum Nevertheless, the blastomeres which results from the first cleavage are unequal in size, and the cellular descending of one these blastomeres divide faster than the descending of the other blastomeres. Descendants of the other blastomeres. Some conditioning effects must be present in the egg cytoplasm, which determines the size of the blastomeres and the rate of later cleavages. This apparent fact suggests that the egg in its development is a builder which lays one stone here, another there each of which placed with the reference to further development. E.R. Lilly in 1895. Next, chemical changes or the physiology of cleavage. Chemical changes or physiology of cleavage. Which is the chemical changes at the time of cleavage and physiology changes also. The cleavage is a phase of intensified chemical activity and transformation of molecule. Besides the energy yielding catabolic activities, the anabolic activities provide to the dividing egg a continuous supply of raw materials, for the biosynthesis of macromolecules of DNA, RNA, proteins, lipids, and polysaccharides, which are required by the cleaving egg for the mitosis. Increase of nuclear material. Number one, increase of nuclear material. The most important changes during the cleavage is the steady increase of nuclear material at the expense of cytoplasm. The number of the nuclei is of course double with every new division of the blastomeres, and this increase is accompanied by an increase in nuclear substance, which involving the DNA. These are several possible sources. Such material Pratchett in 1950 by the study of the sea urchin egg. Next, ribonucleic acid synthesis. During early cleavage, chromosomal DNA remains busy in the own duplication and exists in an active state. Therefore, no or little transcription of any kinds of RNA during the period of early cleavage occurs. The nucleolus is the site of ribosomal RNA, but this organoid is completely absent in the egg of frog and sea urchin during the cleavage. This description described the scientist Brown in 1966. Hence, no ribosomal RNA is produced. Third number, protein synthesis. It has been noted that there is a spectacular increase in protein synthesis, and this is the continued throughout the period of cleavage. If the cleaving eggs are treated with puromycin, which inhibits RNA, depends protein synthesis. Cleavage stops immediately, thus showing that protein synthesis is indispensable for the cleavage to take place. Okay, thank you.